plant breeding section, crop science division, ICAR RC region, Umiam, Meghalaya. So, dear students, we have a great opportunity to esteemed scientist, Dr. Philanim, and she will be delivering on the topic of plant science. So it is mostly useful for horticulture and agriculture, and the students who are pursuing for JRF, going to appear for JRF examination. So it will be more useful for you. Be attentive and make use of the time, because our university is providing a good platform to invite the learned resource person, and they are in their respective field, and bringing them in one platform to educate you and train you for this exam. Make use of this time, and if you have any queries and questions, you can write it on the chat box, and also at the end you can interact with her. Okay, so may I request now Dr. Philanim to take the time and start your lecture, please.
Okay, thank you so much, sir, for your kind introduction, and good morning, everybody. I'm Dr. Philanim, and I'm working currently as a scientist at ICAR Regional Center for Northeastern Hill Region, Umiam, Meghalaya. So my department is plant breeding and genetics. So I will be spending time with you today to discuss and to give some tips and suggestions on how to prepare for JRF, especially for the plant science section. So yeah, sir, shall I share the screen?
You can start sharing the screen. No problem.
Okay. Is the screen visible to everybody?
Yeah, visible.
Yes, ma'am.
Okay.
You can make it slideshow view.
Yes, sir. Okay. So the major subject I'll be dealing with will be plant science today. So are all the students planning to write their JRF exam on plant science, or are they here for like just to gain some knowledge?
I think horticulture, agriculture students are there.
Okay. So all the ICR conducts all Indian entrance exam for admission every year for admission to UT, PG, and PhD courses. So I think because of the current pandemic situation, the application has been delayed again. So earlier it was supposed to be the registration, online registration was supposed to be completed by March, but I think now they have delayed it to May and June, if I'm not mistaken. Okay, coming to 1 second. Okay, so we know that under plant science, there are 5 major subjects that it encompasses. First is plant breeding and genetics. Second is plant pathology. Third, agriculture, microbiology, or general biology. And fourth is science and technology. Fifth is your plant resources. So the exam will be conducted for 2 hours. Exam will be conducted 2 hours. And the total number of questions will be 160. Total marks that is given is 640. So for every correct marking, you will get 4 marks. And for every correct mark, you will get -1 mark. Okay, so for unattempted question, there will be no marks. It will be 0. Okay, this syllabus you will already be getting from the site, also from ICAR website, but I've just kept it for your information's sake. So for syllabus in plant breeding genetics, you will have to study the history of genetics, the theories and scientists that are associated with it. So all this in this particular topic, you will have to study all the scientists and the theories and the terms that are associated with its pre-Mendelian, Mendelian, and post-Mendelian era. Okay, so this is very important because during exam they used to ask most of the questions from terms, and especially like for example, like who has coined the term linkage, or who has coined the term heterozygosity. So this kind of questions usually comes up, and you need to remember the various theories and the scientists that have discovered those theories. Okay. Then important terminologies like hereditary traits, genetics, homozygosity, heterozygosity, then linkage, recombination, pseudoallele, codon, pleiotropy, hybrid, cistron, muton, recon. Okay, all these terms you need to remember. And Mendel's law of inheritance. We know that there are 3 laws: laws of segregation, laws of independent assortment, and laws of dominance. Okay, so these laws you need to remember, accept to these laws. So while going through your studying part and while you read through the textbook, try to focus on this. Okay, so one suggestion is first stick to the syllabus. Don't go beyond the syllabus, and try to revise as much as you. Okay, so I think that being delayed, your application getting delayed and your exam getting delayed because of the current situation is a blessing in disguise, because you have more time to prepare for the exam. Okay, so prepare in such a way that, because this is JRF exam is an objective based exam, so you did not go into deep theoretical lesson and try and remember every detail of each topic. Okay, you can just, how you can study, you can do smart reading, like whatever important terms or whatever important concept that is associated with a particular topic, remember that. You don't have to remember every detail of each topic. Okay, and revision is key. So first of all, you stick to the topic, stick to the syllabus, and keep revising. Okay, if you want, you can make your own notes also. So keep revising, because while reading it, you may find it very simple, and it's very, you may feel that, okay, this is easy to remember and I can remember it in the examination hall. But during the examination, what happens is even the simplest of simplest, it gets confusing. So that is why it is so important to, that's why we keep suggesting students also to keep revising, because in the exam they may just ask which is a cross-pollinated species. Okay, and the option might be wheat, maize, rice, and black gram. Okay, and also sometimes we get confused. Some students go for wheat and some students go for black gram. Okay, so these things, even though it is very simple, you need to keep revising it, so that it'll be ingrained in your brain and you will not forget it. Okay, and stick to the syllabus. Of course, later on, if you have extra time, then you can definitely venture yourself into other topics or other latest developments in genetics and plant breeding and in other subjects. Okay, so you can go into that, but first stick to the syllabus. So then you have your linkage and crossing over, chromosome structure, number, karyotype, ideogram. Okay, I will not go and read each and every topic, that will be exhausting. And anyway, when you're going to syllabus, you will also know by yourself, like what to read, what not to read. Okay, here I mentioned as the based on where the chromosome is, based on where the centromere lies in the chromosome. Okay, when you're karyotyping it, when you're seeing it in the microscope, you will be able to know that a different shape of chromosome. Okay, for example, if your centromere is lying in the center of the chromosome, it will give you a V-shape chromosome. Okay, if it is lying a little away from the centromere and towards the telomere, then it will form an L shape. It is when it is very near to the telomere, okay, it will give you a J shape, which is when your centromere is exactly adjacent to the telomere, or it is very near to the telomere, then it will give you an I shape. Okay, so these things, because I have when I was going through the past question papers, these questions were often asked. That's why I have included this specifically. So try and remember the centromere position, the names given to it, and the shape that you will be able to karyotype when you see it. Okay, so you need to remember these things. And yeah, the rest of the syllabus I will not go through. This codon, anticodon, and genetic code we will be discussing later on. Okay, then the gene expression, lag operand, fine structure of a gene. Okay, in plant breeding, there are different syllabus, different topics that are given. So you may go through according to the syllabus. Cytogenetics. Cytogenetics is also equally important, especially your chromosomal aberration part, where you're dealing with low polyploidy or polyploidy, and your different kind of numerical aberrations and structural aberrations. Okay, so this is especially very important because every year they have asked questions from this particular topic. So while studying, this stress more into these topics. And biometrical also, it is important to know every significant statistical test and why we use those statistical tests. Okay, for example, chi-square test, F test, T test, Z test. In what situation do we use this in the field, and why do we use it? In different kinds of experimental designs, like your complete randomized design, randomized block design, LSD, okay, augmented block design. So all these kinds of design you will have to remember. You don't need to study the layout of the design, or you don't need to remember every detail of the design. You just have to remember what design it is and where it is used. Okay, so these two things you need to remember when you're studying the experimental designs. Okay, coming to the plant pathology also, this is the syllabus. I will not delve into plant pathology and other subjects because that is not my area, but I will, of course, I will give you tips on that, and will also give study material. What are the books that you need to study specifically for these subjects? Okay. Seed science and technology, microbiology, and plant genetic resources. Like I said, these 5 subjects, plant breeding and genetics, plant pathology, seed science and technology, microbiology, and plant genetic resources. These are the 5 core subjects of plant science. Okay. So, but apart from this, every year, like a subject, ask questions from crop physiology and intellectual property rights also. You need to focus on these topics also. So in crop physiology, like I have said before, no need to study the entire textbook. There are certain topics which are often asked. So I have listed those topics for you. So you may go through these topics. And intellectual property rights also, there are certain topics. So please go through these topics. Okay. Okay, I will come back to this later. Okay, now we will discuss something on genetic code. I have picked a few topics for today's class because these topics are very important when we are talking about genetics and plant breeding, and the questions have often been asked from these topics. Okay, so of course, you'll be asked questions from plant pathology and from microbiology, from other sections also. But from when you're talking about plant breeding and genetics, they will often ask questions from these topics. And if later on, if we can have a second class or something, then I will include other topics also. Okay. So genetic code. Okay, genetic code, as you know, that it is a sequence of DNA or RNA that codes for a particular or a specific amino acid, okay, in the protein. So, as you know, codon is the number and the sequences of the bases in the mRNA that specifies for a particular amino acid. So, as you know, codon will have 3 nucleotides. Okay, so first of all, in mRNA, you have what are the 4 mRNA nucleotides? They are adenine, thymine, guanine, and cytosine. Okay, and then in tRNA, instead of thymine, you will have uracil. Okay, so it will be adenine, uracil, guanine, and cytosine. So remember this. Okay, and codon will be those 3 numbers. Okay, the number and the sequence. So the number will be 3. It will be either of the 3 nucleotides of ADGC. Okay, and the sequence might be in any format. It could be ADG, it could be TGC, it could be AGC, any sequence. Okay, so that sequence of mRNA that codes for a particular sets of anticodon in the tRNA. That is your codon. Okay. And anticodon, of course, it's the set of bases in the tRNA that pairs with the codon of an mRNA. Okay, so genetic code is all the sets of the codon. That is the 64 sets of codon that specifies your 20 amino acids. Okay, so we know that there are 20 amino acids that our body needs. Okay, and 9 of these amino acids are essential amino acids. Okay, so this 64, the entirety of 64 codons, they will code for this 20 amino acids. Okay. So start codons, as you know, the start codon is AUG. AUG is methionine, and your stop codon is UAA, UAG, and UGA. Okay. The name that is given to each of these stop codon or the nonsense codon are for UAA, it is ochre, for UAG, it is amber, and for UGA, it is opal. So they are nothing but they are color schemes. Okay, they are just colors. Ochre, amber, and opal are name of colors. So the scientists these 2 scientists who discovered this stop codon, they gave the name amber. They gave the name amber. Amber was one of the nicknames of one of the scientists. Okay. So they from there, this amber name has derived. Ochre and opal, they have just succinctly they have included subsequently later on to make it a color scheme. Okay, so ochre, amber, and opal. They might ask you like UAA is, you know, what is the name of the stop codon UAA? So you have to take ochre. Okay, so you need to remember the name of this, the name of each of this stop codon. As you know, start codon is something that it is a part of a gene where your RNA polymerase, okay, RNA polymerase will come and bind to this particular start section so that your transcription will initiate. And stop codon is the termination point, okay, the termination point of your transcription. Okay, so some of the important characteristics of genetic code. The first is non-overlapping. Non-overlapping means no two mRNA, the same mRNA will not occur in more than 1 codon. Okay, that means, for example, non-overlapping means if CAT codes for an amino acid, say cysteine, then the next codon will not be ADG. Okay, it will not be, or it will not be TGA, because CAT is already occupied by cysteine. Okay, so the next codon will be GAD. Okay, so it will be separate. So they will be non-overlapping nature of the codons. Commaless means there will be no punctuation mark in between. So no codon will act as a punctuation mark. See, for example, if CAT is one of the codon that codes for cysteine, then there will not be a codon here, like for example A or T or G, that will act as a comma or a punctuation mark or a full stop. Okay, so there's no comma, no punctuation mark. Okay, it is a continuous flow of codons. The third character of a codon is genetic code is unambiguousness. Okay, that means unambiguousness means each codon specifies for only 1 amino acid. Okay, so each codon specifies for only 1 amino acid. That means if you see the example here, UUU will specify only phenylalanine. Okay, you do not see in any other protein or any other amino acid where UUU is coding for another amino acid. Okay, UUU only codes for phenylalanine and does not code for any other amino acid. Okay, so that is what unambiguousness means. But there are exceptions to this. For example, in AUG in prokaryotes, chloroplast and mitochondria, they code for methionine, whereas AUG in eukaryote will code for methionine. Okay, so AUG in prokaryote, chloroplast and methionine codes for methionine, whereas AUG codes for methionine in the eukaryotes. The fourth is universality. Okay, universality of the codon means a codon codes for the same amino acids in all the organism. So if UCC is coding for serine in maize, then UCC will code also for serine in rice. Okay, so the same codon will code for the same amino acid in different organisms or different species. Okay, so that is what is universality. And eukaryotes, in eukaryotes, but there are again exception to this. So isoleucine in eukaryotes is coded by AUA, but in E. coli, isoleucine is coded by AUU and AUC. Again, in some celiacs, UAA and UAG, which is a stop codon, codes for glutamate instead of termination codons. Okay, so there are exceptions to the universality also. The fourth is the degeneracy. Okay, so degeneracy means one amino acid might be coded by more than one codon. Okay, so what this degeneracy means one amino acid might be coded by more than one codon. See, for example, in phenylalanine, it is coded by two codons, UUU and UUC. Okay, same wise for arginine. It is coded by CGU, CGC, CGA, and CGG. So arginine is coded by 4 codons. Okay, so that is what degeneracy means. Degeneracy means a single amino acid might be coded by more than one codon. Okay, so that is what degeneracy means. But again, there are exceptions to this. Like AUG codes only for methionine. It does not code for any other. Methionine is coded only by AUG. Okay, AUG does not code for any other this thing. Okay, and then UGG, tryptophan is coded only by UGG. Okay, there are no other codon that codes for tryptophan. Now, this degeneracy nature of codon is because of something called as the Wubbel hypothesis. Now, you have all heard what Wubbel hypothesis is. You might have already read, or the teacher might already have explained to you about this in the class also. So Wubbel hypothesis was discovered by Crick in 1966. Okay, so in what Wubbel hypothesis says is the first two the first two pairs of a codon, okay, they follow strict pairing rules. Okay, stringent pairing rules. So stringent pairing rules means, what is the stringent pairing rule of a nucleotide? It means A will always pair with T, G will always pair with C. Okay, so this is the stringent pairing rule. So they will not deviate from the norms or the rules. Okay, but the third base in the codon and the first base in the anticodon, they do not follow this rule. Okay, so they are wobbly in their base pairing nature. So the third base in the mRNA and the first base in the tRNA will code for example, the uracil will code for adenine or guanine. Guanine will code for cytosine or uracil. Ionic acid will code for uracil, cytosine, and adenine. Okay, so they do not follow the strict pairing rules. Okay, so because of this, degeneracy happens. Because of this, one amino acid is coded by more than one codon. Okay, so if you see, you will see the degeneration nature only in the third codon. For example, in phenylalanine, the first two pairing will be stringent. Okay, only the third codon will change. Okay, only the third nucleotide will change. So this kind of situation is created because of the Whipple hypothesis. Okay, am I audible?
Yes, ma'am.
Yes, audible. You can continue.
Okay, okay. Because it was showing that my network is not good like that, no? So I just wanted to confirm. Okay, the next is chromosomal aberration. Okay, chromosomal aberration, you have two kinds: structural aberration and numerical aberration. Okay, so structural aberration encompasses deletion, duplication, inversion, and translocation. So what deletion means, as the name itself indicates, deletion means a particular segment of a DNA will get omitted out from the chromosome. Okay, so there are kinds of deletion. The first is the terminal deletion. Terminal deletion means your removal of the segment of the DNA will occur towards the end of the chromosome. That means it will include the telomere as well. Okay, but this is very uncommon because telomere, as you know, is necessary for to maintain the stability of the chromosome structure. So this is very uncommon. Okay, and the interstitial deletion is a segment between the telomere and the centromere will get deleted. So they usually ask questions like how many breaks are there, how many breaks are required in a terminal deletion, or how many breaks are required in interstitial or intercalary deletion. So you have to remember that in terminal deletion, there will be single break. Okay, there will be single break because your deletion will occur somewhere at the end. So there will be only one break. And in interstitial deletion, there will be two breaks because your deletion is happening between the centromere and the telomere. Okay, so there will be two breaks. And yeah, so you need to remember this. And the genetic effects or the genetic imbalance that it will cause is most deficiency will act as recessive letals. Okay, recessive letals means they will only kill the organism or cause some kind of instability to the organism if there are two copies of the lethal gene. Okay, two copies of the alleles. Okay, if there is only a single copy of the allele, then the organism will be normal. Okay, the carrier will be normal. But if there are two copies of the lethal gene, then it will cause the death of the organism, or it will cause some kind of instability or abnormality in the organism. So that is the recessive lethal. Okay, large kind of deficiency will produce dominant lethal gene. Dominant lethal gene means single allele is enough to kill that organism or to cause some kind of instability. Okay, so example for this is Huntington disease. Okay, so most of the deficiency will cause your recessive lethals. If the segment that is removed is large enough, it will cause dominant lethal. It will cause the expression of dominant lethal genes. Okay, pseudodominance. Pseudodominance means there are conditions where the recessive allele, okay, if it is present in the homolog counterpart of the deleted segment, okay, then if the dominant allele gets omitted out, then the recessive allele will express its trait. So this is pseudodominance. Okay, as long as the deleted portion is there, present, and if the deletion does not occur, then this recessive allele will not express. But if in its homolog the deleted portion has dominant allele that gets omitted out, then the recessive allele will act as a dominant allele and it will express. Okay, so this is pseudodominance. Okay, and the crossing over will be absent in the deleted segment. Okay, how it will be visible in the during the pairing, during the Peckytian stage. Okay, so this is how the chromosome, how this abnormality will be viewed during the Peckytian stage. So in interstitial, during the Peckytian stage, it will form a loop. Okay, it will form a loop for the recombination or the crossing over to happen. So, for example, here, A, B, C, D, E, F. Okay, this is the alignment or the series of alleles that are present in one homolog chromosome. Okay, and this same representation is also present in the other homologue. Okay, but in the other homologue, your CD sequence gets omitted out or deleted. Okay, so what will remain? A, B, E, F will remain. So during synapsis, what will happen? The alignment will be the same. Okay, they will align towards each other. But because in its homologue, CD is omitted out. Okay, segment is omitted out. So the other homologue, the normal homologue, will form a loop. Okay, it will form a loop so that proper alignment of AA, BB, EE, and FF can happen. Okay, so for this, for proper crossing over to happen, okay, to avoid any kind of error in the recombination, the normal homologue will form a loop for during synapsis. Okay, so that is why when you view the chromosome during the pectitin stage of the prophase, you will see kind of a loop formation. Okay, in the deleted segment. In terminal segment, the remaining part will, the normal chromosome will have the original sequence of A, B, C, D, E, F, but its deleted homologue. Okay, it will have the end portion will remain unpaired. Okay. Some of the example of diseases that are caused because of deficiency is Philadelphia 22 and Trisomy. Philadelphia 22 is a deletion into chromosome number 22, and Trisomy is a deletion that is found in chromosome number 5. Okay. Excuse me. Next is duplication. Duplication, as the name itself again indicates, it is presence of extra segment chromosome. That means one segment of chromosome will be represented 2 times. So in duplication, there are 4 types. Tandem duplication. Tandem duplication means your duplicated segment will lie right next to the your normal segment. Okay, so A, B, C, D, C, D, E. Okay, C, D, E is duplicated. So the extra segment will lie next to CDE. Okay, that is your original segment. So this is tandem duplication. Reverse tandem duplication means your extra segment will lie next to your original segment, but in reverse order. So CDE is your original segment, and EDC will be in the reverse order, and it will lie next to CDE. Okay. Then displaced duplication means displaced duplication. The original segment, the extra segment will move away from the original segment. Okay, so if your original segment is CDE, your extra segment is near your centromere, and in between the original segment and your copied segment, there are the alleles present as well. Okay, like FG. And your translocated duplication means the extra segment is a non-homologous chromosome. Okay, so for example here, this is another non-homologue. Okay, in this, the segment from another non-homologue CDE has got transferred and integrated. Okay, so this is your translocated duplication. So duplication might occur because of, most commonly because of non-allelic homologous recombination. That means usually when the chromosomes align during pectitin for crossing over, they align allele to allele. Okay, so allele in the one copy of the allele in one homologue will align with another copy of the allele in another homologue. Okay, so capital A and small a will align, capital P and small p will align, and that's how recombination will happen. Okay, but sometimes this kind of alignment is not there. Okay, sometimes alignment happens between the non-alleles. That means C and D will align with small a and small b. So they are no doubt they are same homologous chromosomes. Okay, they are homologous chromosomes, but they are non-allelic. Okay, so because of this non-allelic homologous recombination, it will lead to deletion and duplication. So after crossing over A, B, C, B, C, and D, so BC is getting duplicated. Okay, and AD may the other segments are getting deleted. So this is one of the major cause why duplication occurs. Okay. Duplication can be within homologous chromosomes. It can be within a single chromosome. It can be within a single chromatid also. Okay. So it is shown here. Again, in duplication also, you will see a loop formation. Okay. Loop formation. Your duplicated portion will form a loop so that normal pairing can happen and recombination can happen. Okay. Coming to the cytology, yeah, I have told you, in Pectinian stage, there will be loop formation for normal pairing. And the genetic effects of this duplication will be bar-eye duplication, okay, the next is inversion. Inversion is your segment, your normal chromosome segment gets inverted 180 degrees. Okay, so for example, this is your normal chromosome. Okay, A, B, C, D, E, F, H, I, J, K, L. So it gets inverted. So original segment will be CDE, gets inverted into EDC. Okay, so there are two kinds of inversion, paracentric and pericentric. Okay, paracentric is when your inversion does not occur centromere, or if the inversion happens within the centromere. For example, in the first case, inversion is happening in your long arm, that is your Q arm of the chromosome, but it is within the centromere. Okay, so this is your paracentric inversion. Pericentric inversion means your inversion will involve your centromere. Okay, so in this case, inversion is happening both in your Q arm and your P arm, your shorter arm and your longer arm. Okay, so pericentric inversion will encompass your centromere as well. Cytology will be there will be absence or reduced frequency of the crossing over. Okay, most because of the alignment error. Okay, because of that there will be reduction in crossing over, and you will also notice the inversion loop. Okay, inversion loop is a little different from the inversion loop that you see in duplication and deletion. Okay, in inversion loop, in duplication and deletion, what happens is the duplicated or the deleted homolog, okay, that will form a loop. Okay, in the case of deletion, the normal homolog will form the loop. Okay, in the case of duplicated, your duplicated homolog will form the loop. Okay, whereas in case of inversion, both your normal and your inverted chromosome will form the loop. Okay, as you can see here. So this is all for proper alignment during crossing over. The next is translocation. So translocation means a segment of chromosome will go and get integrated to another non-homologous chromosomes. Okay, so there are three kinds of translocation: shift, simple, and reciprocal. So in simple translocation, the terminal segment of a chromosome will go and integrate with in the end portion of the non-homologous chromosome. Okay. So this is a simple kind of translocation. In this, there will be ABC. ABC will break from its original chromosome, and it will go and integrate with another non-homologous chromosome at the end of the chromosome. Okay. So this is simple translocation. In shift translocation, an intercalary segment. So whenever we say intercalary, you remember that there will be two breaks. Okay. Whenever we say simple, there will be a single break. So in intercalary segment will go and integrate itself in the non-homologous chromosome. Okay. So this is more common and occurs in Drosophila and Neurospora. Okay. So you can see here, sequence DE breaks one from the region near to centromere, one from the region that is towards the telomere. Okay, so there are two breaks, and they will go and integrate intercalarily again in the non-homologous chromosome also, they will not go and integrate in the towards the end. Okay, they will go and integrate in between the centromere and the telomere. Okay, so this is your shift translocation. Reciprocal translocation means the two non-homologous chromosomes will exchange. Okay, they will exchange segments. So again, here also there'll be single break. They will exchange segments. So A, B, C, and I, and will get exchanged between these two non-homologs. Genetic effects. Genetic effects mean there will be alteration in the genetic linkage map of the affected chromosome. So when you say linkage map means all the alleles, all the genes that are present in a single chromosome, that is your linkage map. Okay. So for example, if I say linkage map of maize, so the total number of chromosome of maize is 20. Okay, so 2n is equal to 2x is equal to 20 in maize. Okay, so linkage map for maize means it will be 10. Okay, that means single alleles, okay, present in single chromosome. So you know that every allele will have a complement in its homolog. Okay, so we are not taking into account the alleles that are present in the homolog, only the alleles that are present in a single chromosome. That is your linkage map. Okay, so the translocation will affect your linkage map. That means, for example, in this case, if you see the original linkage map that it has is 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, let's say original linkage number was 9 in this case. But because the terminal section got removed and it got translocated to another non-homologous chromosome, now the linkage map becomes 1, 2, 3, 4, 5, 6. Linkage map number of this chromosome becomes 6, whereas the linkage map number of this chromosome becomes, say, 12. Okay, the non-homolog becomes 12. So this is how it will change the linkage map of the affected chromosome. Okay. Chromosome pairing, it will show a T-shaped pairing. Okay, so you need to remember all this, which shows loop formation during the pectine stage. Okay, what kind of configuration will be seen during translocation, what kind of shape it will show, what kind of ring formation it will show. So these things you need to remember. Okay, they might ask questions on this. And chromosome pairing means T-shaped pairing during pectine in translocated heterozygote. So translocated heterozygote means one pair will be normal and the other it will be translocated. Okay, so it will form a T-shape pairing for the crossing over to happen. So wherever there is complementary, they will pair. Okay, so here you have the normal homolog. Opposite to it, you have the other normal non-homolog. Okay, and then adjacent to this, each of their homologs are present adjacent to this. Okay, so they form a T-shaped configuration for normal crossing to occur. Okay. Now coming to chromosome disjunction. Chromosome disjunction means after they have after the crossing over is over and after they have aligned themselves in the metaphase plate, how they will move away from the center towards the pole of the cell. Okay, so there are three kinds: adjacent 1, adjacent 2, and alternate. So in adjacent 1, the two non-homologous centromeres lying adjacent to each other move to the same pole. So for example, this is chromosome, normal chromosome from one homolog, and this is normal chromosome from other homolog. Okay, but this has got translocated. So these two will move together to the same pole, and these two will move together to the same pole. Okay, but the product will be inviable because if you see this condition, one is normal but one is not. Okay, one is translocated. In this condition also, one is normal and one is translocated. So there will be deletion or duplication that occurs, okay, during the progeny formation. So because of this, the product will be inviable. The second is the adjacent 2 segregation. So in adjacent 2 segregation, the two homologous centromere lying adjacent to each other move to the same pole. So they are homologous. Okay, they are homologous except that this homologue has a translocated segment from other non-homologous chromosome. Okay, so these two homologues will move to the same pole, and these two homologues will move to another pole. Okay, so in this, again, in this condition also, gametes will be inviable because there will be deletion and duplication in the progeny. Okay. The third is your alternate segregation. Alternate segregation means the centromere lying alternate to each other in the ring move to the pole. That means the two translocated chromosomes will move to one pole, and the two normal chromosomes will move to the other pole. Okay. So in this case, you'll get viable gametes because except for the translocated ones, the chromosomes that have moved to the other pole, they are completely normal. Okay. So you'll get viable gametes in this case. Okay, now there is something known as, there's a concept known as centric fusion or Robertsonian translocation. Okay, Robertsonian translocation means there will be translocation, reciprocal translocation happening between two acrocentric chromosomes. Okay, so acrocentric chromosome is where the centromere lies closer to the telomere. Okay, so between these two non-homologous chromosomes, if there are translocation happening, then it will give rise to a microchromosome and a metacentric chromosome. Okay, so let's say there's a break here and there's a break here in the second chromosome. Okay, so these two chromosomes will get integrated and these two chromosomes will get integrated. Okay, and because the microchromosome, the area near the centromere are mostly heterotic, the microchromosome cannot survive. Okay, so in the future generation, this chromosome slowly gets lost. So it will give rise to the formation of a metacentric chromosome. Okay, the reverse process. Okay, the reverse process where there's a translocation between a microchromosome and an metacentric chromosome is called dissociation. Now, the next kind of structural aberration, chromosomal aberration, is the numerical aberration. So in numerical aberration, it will be a deviation from the diploid chromosomal state. That means there will be gain or loss of few of the chromosomes. Okay. In structural aberration, there is also loss of segment of DNA. It is just a segment of a DNA, not the entire chromosome. In numerical aberration, there will be loss of the entire chromosome. Okay, one or two chromosomes. Okay, so in this, first is nullisomy. So nullisomy means an entire chromosomal pair will get lost. Okay, so one homologous chromosomes will get lost. That means 2n minus 2. So for example, in human beings, we have 46 chromosomes. Okay, so if nullisomy occurs in human beings, then we will have only 44. That individual will only have 44 chromosomes. Okay, that is nullisomy. And because both the chromosomes are getting lost, and the genes that are present in those two chromosomes or the homologous chromosomes could be viable for the survival of the individual. And if those genes gets lost, then the individual will not be able to survive. Okay, that's why most of the diploids, okay, they are not able to survive this. But they are viable in polyploids, for example, in wheat, but there will be reduction in the size and fertility. So in polyploids, as you know that there are more than one genome. Okay, unlike diploid, there are more than one genome, but each genome will have two copies. Okay, so because there are more than one genome, and even if a portion of two chromosomes or two homologous chromosomes gets lost, there could be other homologous segments that could be present in other genomes. So that's how the organism is able to survive, or the species is able to survive. Okay, so but again, the functioning of the plant or the functioning of the organism will differ from crop to crop or from species to species. Okay, in wheat, as we see that there is, even the plant able to survive, there is reduced vigor, there's a and there is reduced productivity. Okay, so in wheat also this has been seen. Then in cytology, cytology will be chromosome pairing will be normal with n-1 bivalence. So n-1 means bivalence means two homologous chromosome pairing together. They are usually termed as bivalence. Okay, so n-1 because there will be one chromosome pair that will be missing. Okay, so the gametes that that will be formed will be n-1. Okay, the next aberration that we see is monosomy. So in monosomy, not a chromosome pair, but a single chromosome will be missing. Okay, so again, it will be inviable in diploids, but it will be fully viable polyploids like wheat, cotton, oats, and tobacco. Okay, so usually they ask questions from here. So complete sets of monosomics are available in which crop? Okay, so you have to list any of the crops that are given here. Say they might in the option they might give you wheat or cotton or oats or tobacco. Okay, so if they ask you complete sets of monosomics are available in which crop, then you have to remember these options. Okay, and even though maize is again a monosomy, again a diploid plant, but maize has a complete set of 10 sets of monosomic. Okay, so again, the performance of the plant will be weak, the productivity will be less. So you need to remember, so they may ask you complete sets of monosomic that is available or complete sets of monosomic that which diploid has a complete set of monosomic. So you need to remember the option. Okay, it'll be maize. Then cytology, again n-1 bivalence, and monosomic chromosome is present as a univalent. Okay, univalent because the chromosome will not have any other homologue to pair with because its homologue will be missing. Okay, now from this we have a particular concept known as univalent shift. Univalent shift means some progeny of a monosomic, for example, in the mother parent, if the mother parent is monosomic for chromosome number 5, then the progeny is monosomic for chromosome number 7. And this is because sometimes more than one univalence are formed. Sometimes because of some error, all the chromosomes are not able to perfectly pair with homologue. Okay, so the univalent will be there. The univalent of say chromosome number 5 that observed in the parent will also be there, present in the progeny. Apart from that, there will also be other univalents that are formed. Okay, because of incompatible pairing or because of unpairing of the other homologues. The next is trisomy. Trisomy means, unlike monosomy, trisomy means you will have another extra chromosome. Okay, so example for this is Down syndrome. In Down syndrome, you have 21 chromosome trisomy. That means your 21st chromosome has one extra chromosome. Okay, so the Down syndrome is also known as mongoloid syndrome because people usually the features change as those of a mongoloid because our eyes become their eyes become smaller, or the nose become flatter and things like that. Okay, that is why it is known as mongoloid syndrome also. And there is mental retardation, there is a speech impairment. Okay, so all of these symptoms are there in Down syndrome, and this is because of the trisomy in 21st chromosome. Okay, now trisomies, there are primary trisomics, secondary trisomics, and the tertiary trisomics. Okay, so primary trisomics means the additional chromosome is an altered member of the haploid chromosome. Okay, so the extra chromosome that is present is the exact copy of the normal chromosome. Okay, so there are two pairs of normal chromosome, there are one homologue. The extra chromosome will be the exact identical of this homologue. Okay, so that is your primary trisomics. So for example, here also they frequently ask questions like how many primary trisomics are possible in maize. Okay, so maize has total chromosome number of 20. So the number of primary trisomics that will be possible will be 10. Okay, obviously because you will have one extra pair of each chromosome. Okay, that is the number of possibility. So if they ask you how many primary trisomics is possible for barley, but chromosome number of barley is 14. So you will say that number of primary trisomies that is possible in barley is 7. Okay. Then secondary trisomies. In secondary trisomies, unlike the primary trisomies, the extra chromosome that is present is an isochrome. Isochrome means both the arms will have the same gene content. Okay, so how isochromes are formed is there will be a transverse centromeric division. It is not a longitudinal centromeric division. It is a transverse centromeric division. And therefore, this arm and this arm will become one chromosome, and the shorter arms, your P arms, will become one chromosome. Okay, so that is why we say both the arms have the same gene content. In this case also, in the second chromosome that is formed also, both the arms will have the same gene content. Okay, so here also they usually ask questions like how many secondary trisomies are possible in barley or in maize or in wheat or in another crop. Okay, then you have to say that because each chromosome, okay, when they pair, they are producing 2 isochromes. Okay, you remember this. Each homologue, when they undergo transverse centromeric division, they are producing 2 isochromes. Okay, or 2 isochromosomes. So your answer will be, for example, how many secondary trisomies is possible in barley? Your answer will be 14. Okay, because 2 isochromes are formed. Okay, if they ask you how many secondary trisomies are possible in rice, then you will say it will be 24. Okay. The next is your tertiary trisomies. In tertiary trisomies, the extra chromosome is a translocated one. Okay, so the genetic effects that will be present, or the genetic imbalance that we can see will be this. Unlike your monosomy and your nullisomy, trisomies is viable in diploids. Okay, because you are not omitting out. There is no omission of a complete chromosome. There is only addition of extra chromosome. Okay, so there might be enhanced or reduced performance, but there will not be it will not be completely lethal for the organism. Okay, full sets of N primary trisomies are available in maize, tomato, in pea, barley, and bajra. Okay, and the performance varies from crop to crop. Okay, now coming to the cytology. In cytology, primary and secondary trisomies will form a trivalent. Okay, it might form a trivalent. That means the 3 chromosomes might pair together because they are homologous to each other. Okay, and again in the primary and secondary trisomies, because the third chromosome is a similar copy of the other homologous chromosome, so they will form a trivalent. Okay, or a bivalent and a univalent. A bivalent means the original 2 homologous chromosomes will pair, but the extra third chromosome will remain unpaired. Okay. Then the secondary trisomic, a very interesting to notice is that secondary trisomics will form a ring-like configuration in the metaphase 1. Okay, so 1 and 2 is your original homologous chromosome, and 1 and 1 is your isochrome. 1 and 1 representation. Why? Because they have the similar gene content. Okay, so similarly 1 and 2 is your original homologous chromosome, and 2 and 2 is your isochrome. 2 and 2, why? Again, because of the similar gene content. So they will form a ring-like configuration during the metaphase 1 for recombination to happen. Okay, not recombination to happen in the metaphase plate. Okay, in the metaphase plate, before they segregate to different poles, this is how this kind of configuration is what you will see. Okay, the ring-like formation. In tertiary trisomics, dumbbell shape formation. Dumbbell now, so this dumbbell formation will be there in the tertiary trisomy. So dumbbell is nothing a pentavalent. A pentavalent means 5 homologous chromosomes will configure, or they will pair together. Okay, so 1 and 2 is one homologous chromosome. 3 and 4 is another homologous chromosome, but 1 and 2 and 4 are homologous chromosomes. Okay, and 1 and 4 because this has a segment of chromosome from 1 and 2 and has a segment of chromosome from 3 and 4. So these 5 chromosomes will pair together to form a dumbbell shape configuration in the metaphase 1. Okay. Next is your tetrasomy. Tetrasomy means there is extra homologous pair of chromosome along with your somatic chromosome. Okay. This is more deleterious or lethal than the diploid. Normal tetrasomics is viable in polyploids. Okay, example wheat. Again, how they will recombine? Again, what kind of recombination they will show is they can either show a quadrivalent. Okay, that means all the 4 both the two homologs. Okay, both the 2 homologs may pair together to form a quadrivalent or a trivalent. Okay, a trivalent and a univalent. That means 3 chromosomes will pair, leaving the third one unpaired. Okay, or 2 bivalents. That is 2 homologous, one homolog and another homolog. They will pair together to form 2 bivalents. Okay, now. Next is euploidy. Euploidy means there will be presence of 1 or more complete genome of an organism. Okay, it could be from a different species also. So unlike aneuploidy, where you have extra set of 1 or 2 chromosomes, in euploidy you have an entire genome present. Okay, so here usually students gets confused. Okay, usually student gets confused here between monoploidy and haploidy. Monoploidy means it is a haploid chromosome number of a diploid. So when you talk about monoploid, it is specifically assigned to diploid organism. Okay, so it is a single copy of a single genome. Haploidy means it is a gametic chromosome number of species, irrespective of whether it is a diploid or a polyploid. Gametic chromosome number means it is half of the total chromosome number that is present in a diploid or a polyploid species. Okay. Polyploidy means presence of more than 2 genomes in an individual, like we've already discussed. And again, in polyploidy, there are 2 types: autopolyploidy and allopolyploidy. Okay, so in autopolyploidy, the extra chromosomes that are present will be identical. Okay, so that will be a chromosome doubling from the same individual or from the same species. Okay, so you have different types: triploids, tetraploids, pentaploid, hexaploid, heptaploid, and octaploid. Okay, so chromosome, how they will be produced? Chromosome doubling may occur simultaneously in the somatic cells that may give rise to tetraploids, or your gametes might be 2n instead of n, okay, which will give rise to autopolyploidy spontaneously. Some kind of heat or cold treatment or irradiation of the seeds with the X-ray or gamma rays, okay, and the plant regenerated from the tissue culture. But the most successful method and the most widely used method is colchigen treatment, okay. So colchigen treatment, as you know, is specified by its formula C22H25O6N. Okay, so it is an alkaloid that is extracted from bulb of colchigen autumnal. Okay, so it interferes with the development of spindle fibers. So if the chromosomes that are supposed to align in the metaphase plate during your prophase, they will not metaphase plate during your first cell cycle, they will not align. Okay, so because of this misalignment, or because of complete stoppage of this spindle apparatus, the chromosomes will not be able to align, and therefore the whole of your chromosomes, okay, whole of your chromosomes, the homologs, the non-homologs, all your chromosomes will move to one pole only. Usually when we align the metaphase in the central plate, half of the chromosome move to one pole and the other half will move to the other pole. Okay, but in this case, because the alignment is tempered, so whole of your chromosome, okay, whole of your chromosome will move to one pole. So because of this, the chromosome will be doubled. Okay, genetic effects will be Gigantism, that means increase in the general vigor and size. The leaves are larger, they are thicker. The flower and the fruit seeds, the size are much bigger than your normal diploid plant. But again, the number of the fruits, number of the seeds, they will be reduced. Okay, and the cells, the pollen grains, the stomata are larger. They show variable sterility. Okay. They'll be late flowering, late in maturity, so their crop duration will be longer than your normal diploid plant. Fresh weight is more, but dry weight is comparable or lower than the diploids. Okay. Coming to the cytology, multivalence, they can form a trivalent, a bivalent, or a univalent. Okay. Triploids will produce trisomics and double trisomic. Double trisomics means their how it will be represented is 2n plus 1 plus 1. Okay, plus 1 is for extra chromosome, for the first extra chromosome, and the next plus 1 is for the second extra chromosome. Okay, that's your double trisomics. And most popular examples of the triploids are banana and watermelon. Okay, so triploids, as you know, that they usually lead to sterility. So the triploid banana and watermelons, they will have either undeveloped seeds, rudimentary seeds, or seeds will be absent. Okay, so seedless watermelon or rudimentary seeds of watermelon, they are most commercialized in Japan, and because of the triploid nature of the fruit, their fruit shape and size and everything also it changes. Okay, so this has been extensively commercialized in Japan. Okay, there is a fertile trisomic, which is known as which is found in spinach. Okay, so they may ask you like some example of triploids, then you have to say banana and watermelon. Example of fertile trisomic or fertile trisomic is found in, then you have to remember these crops. Okay, then you have autotetraploids. Autotetraploids, the cytology will be it'll form a quadrivalent. Okay, I've explained to you this. It's from a quadrivalent, trivalent, and a monovalent, or two bivalents at metaphase 1. Okay, so it will be unbalanced and it'll be inviable. Some example of autotetraploids are potato, peanut, coffee, and lucerne. Some examples of triploids will be banana, and hexaploid will be sweet potato. Okay, so commercialization is triploid watermelon, as I've mentioned, and autotetraploid, which is a commercial crop variety. Okay, autotetraploid variety in rice, rye has been released. Okay, double still and tetrapecus. Okay. Okay, the next is allopolyploidy. Allopolyploidy means it contains here also it contains 2 copies of or more than 2 copies of the genome, but the extra copies of the genome are from different species. Okay, so most allopolyploids have 2 copies of each genome and show regular bivalence at M1 if they were essentially diploids. Okay, so allopolyploids, again, they are usually known as amphidiploids. Okay, so amphidiploids because of diploidization. I don't know if you guys have heard this term or not, but this is again an important term. So diploidization means there will be some genes present in an organism that will allow or that will make that organism to form bivalence instead of multivalent, despite it being a allopolyploid. Okay, for example, in wheat, 5B gene is present in PH gene is present in 5B chromosome. Okay, so what this gene will do is it will configure in such a way that it will allow only bivalent formation in the wheat instead of multivalent. Okay, so despite having more than one genome and despite having multiple genomes, chromosomes will align themselves as a bivalent. That is, only 2 homologous chromosomes will pair, not 3, not 4. Okay, so this phenomenon is known as diploidization, and the crops that have this phenomenon, they are usually known as amphidiploids. Amphi means both, right? So both means it behaves as a polyploid because of its genome content, copies of genome content, and it will also behave like a diploid because of its bivalent formation. Okay, so this is known as amphidiploid. Coming to synthetic allopolyploids, and allopolyploidy produced through colchicine treatment. Okay, so wheat, for example, is a natural allopolyploid. Okay, so but there are certain crops or certain species that has been produced by colchicine treatment through chromosome doubling. Okay, so one of the most successful example is triticale. Okay, so triticale is a cross between Triticum tertitum and rye. Okay, rye is a sickle cereal. So the genome composition of Triticum tertitum is AABB and rye is RR. Okay, so when they produced the F1 was completely sterile. Okay, so then they gave the treatment of colchicine and they induced chromosome doubling. And finally, they were able to produce triticale, which is an allohexaploid with genome content of AABP and RR. Okay. Okay. So the genetic outcome might be when you are producing synthetic allopolyploids, the outcome might be desirable or it might not be desirable. Okay, for in the case of triticale, the outcome they got was desirable. Okay, because they wanted to induce yield and quality of wheat along with the stress tolerant nature of rye. Okay, so they were successful in this. They were able to produce a crop, a completely new crop with the yield and the quality of similar to wheat and the hardiness that was similar to rye. Okay, so variety TL419 has been released successfully in India. Okay, and actually triticale was first produced somewhere in the late 1900s in Scotland. Okay, later on they have, over successive years of crossing and developments, they have improved this crop, and the commercial varieties that we see mostly now for triticale is a derived variety. Okay, it's not an original cross of tertitum and rye, but it is a derived variety. That means it is a cross between this triticale and triticale. Okay, so they have developed varieties by these crosses. So this is one of the successful example of synthetic allopolyploidy. Again, there are some failures as well. We can see by one famous example that was done between cabbage and radish. Okay, so they wanted to produce a Raphinoprassica, where they wanted to have the leaf of the cabbage and the roots of the radish. Okay, but the result they got were the leaf of the radish and the roots of the cabbage. So it was totally opposite. Okay, so this experimentation was this cross was a failure. Okay. Okay, now you need to remember some of the origin. Okay, some of the origin of some crops like wheat, cotton, tobacco, and brassica. Okay. So origin of wheat, as you know, it is made from a cross between Triticum monococcum and an unknown diploid species. So this unknown diploid species is still debated upon. Some say that it is Agelop speltoids. Okay, so Agelop speltoids, they say it is a donor of bee genome, but again, this is not conclusive. Then a cross between these two species produced Triticum tertitum, and Triticum tertitum again was crossed with Triticum tauchi, which is the donor of D genome, and finally they were able to produce the our present day bread wheat, that is Triticum aestivum. Okay, which is then again an allohexaploid. So I will not go into the crosses of this tobacco and cotton, Try and remember these crosses. Try and remember their genome content, their genome constitution, and their chromosome number, okay, along with the scientific names. One of the famous crosses that was made are the present-day Brassica family, or the Brassica species, has derived from this cross. Okay, it is also known as the Brassica triangle, or the U triangle of U. Okay, because it was given by Nagaharu Yu in 1935. So Brassica nigra, Brassica oleracea, and Brassica compestris, they are all diploid organisms. Okay, so they have made crosses among these, and they have produced amphidiploids. Okay, Brassica carinata, Brassica juncea, and Brassica napus. Okay, so these amphidiploids are produced. Okay, so try and remember the scientific names, the common names, the chromosome number, and the genome constitution. Okay. Okay, so this is an important cross, so you need to remember this. Okay. Okay. The next is apomixis. So apomixis means seeds develop from embryo without fertilization. Okay. So this is your basic structure of your ovule. Surrounding the embryo sac is your chalaza, nucellus, and integument. Okay, and your embryo sac has 3 antipodal cells, 2 polar nuclei cells, 1 egg cell, and 2 synergids. Okay, so there are different terms that are associated when we talk about apomixis, and they usually they usually ask by the definition of this term, or they will give you a concept from this from the term, and then they will ask you what kind of concept it is. Okay, so you need to remember all this. The first is the adventive embryony. Adventive embryony means there will be no production of the embryo sac, okay, but the embryos will directly develop from the surrounding areas of the vegetative cells of the ovule. Okay, that means it will develop either from chalaza or from nucellus or from the integument. Okay, so this is as adventive embryony. Okay, and this phenomenon is usually found in crops like mango, citrus, and orchids. Okay. Now the next is gametophytic apomixis. So in gametophytic apomixis, embryos will develop without fertilization from the egg cells or other cells of the embryo sacs. Okay, so in this, the embryo will develop without fertilization from the embryo sac, or from the egg cell, or from the embryo from other cells of the embryo sac. For typically in this, the plant will generate from the embryo sac. Okay. So in this, again, there are 2 terms, apospory and diplospory. Apospory means embryo sac. Okay, apospory means some vegetative cells of the ovule develop into unreduced embryo sac. That means some vegetative cell of that is the nucellin, chalaza, or the integument will take part in the production of the unreduced embryo sac. Okay, unreduced embryo sac, that because they undergo only completely omit out meiosis. Okay, so there will be no reduction in the chromosome number. So the vegetative cells will lead to the production of the embryo sac. This is your apospory. Okay, so this is found in orchids, Hieracium, or Melas. Okay. And then diplospory means embryo sac will be developed from the megaspore either by omission of the meiosis or by reduction of the meiosis. Okay, meiosis process after the first meiotic division. So in the diplospory, your embryo sac will develop from the megaspore. Coming to parthenogenesis, parthenogenesis may parthenogenesis means an embryo will develop without fertilization. Okay, so there are two kinds of parthenogenesis. One is corneal and the other is somatic. Corneal means embryo will develop from your egg cell. Okay, this egg cell, and then somatic parthenogenesis means embryo will develop from the other cells apart from the egg cell. Pseudogamy means pollination is necessary for the embryo development, but again the fertilization of the egg cell will not occur. Okay, so pseudogamy also it has two types: corneal if it develops from the egg cell, or somatic if it develops from the embryo cell. Now there is a particular term as double fertilization. Okay, so double fertilization means one male gamete will fuse with the egg cell to form your embryo. Okay, and the other male gamete will fuse with the two polar nuclei to form your endosperm. So this is known as double fertilization. So your embryo will be diploid and your fusion of the two polar nuclei with another male gamete will give rise to endosperm. So your endosperm will be triploid. Okay. Okay, coming to quality seed production. This again, questions have come from this again and again, over and over in the past years. So I have again included this topic for today's class. So quality seed production, as you know, there are 5 types of seed production. I'm not going into and explaining each and everything from each topic because I'm just explaining from your exam point of view. So yeah, there are 5 types of quality seed production. The first is the nucleus seed, your breeder seed, foundation seed, certified seed, truly labeled seed, and there is also something known as registered seed. Okay. So coming to your quality seed, coming to the nucleus seed is the basic of your seed production. That is, this is your initial point from where you will progress the seed production. Okay, so it is maintained by the breeder. It is produced under direct supervision of the plant breeder. Okay, so they usually ask questions like nucleus seed is produced, where is nucleus seed produced, or under which supervision the breeder seed is produced. Okay, so these questions they frequently ask, and you need to remember what kind of what color tags is given to each of the seed. Okay, so the nucleus seed, as I said, it is produced under the direct supervision of the plant breeder, and the genetic purity is 100%. Okay, and the breeder seed it's a seed that is derived from the nucleus seed. Again, the genetic purity for the breeder seed is also 100%, and it is produced in the research institute or university under the supervision of a breeder. Okay, but it will be monitored by the breeder and officers of the seed certification department and NSC. A golden yellow tag indicates a breeder seed. Okay. Then the next is foundation seed. Foundation seed is derived from the breeder seed. It is produced at government farms or private seed companies. Okay, and but again, foundation seed production can also be taken up by the farmers by proper procurement of the breeder seed. Okay, and then this will have a genetic purity of 99.5%. White tag is associated with the foundation seed. Certified seeds, it is derived from the foundation seed, and it is produced by national seed company and state seed company. Okay, and the private seed companies and the farmers. And this will have a genetic purity of 99%, and certified seeds are only produced for the notified varieties. Okay, so it is not produced for the released variety. It is only produced for the notified variety. Azure blue tag is given for the certified seeds. Now the next is truthfully labeled seeds. This does not come under the jurisdiction of the Department of Seed Certification, and this is not tested for its genetic purity. It is only tested for its physical purity and germination and moisture content. Okay, and any farmers can produce this TFL seeds, and here the certification is not compulsory, it is voluntary, but the labeling is made compulsory. Okay, so truthfully labeled seeds is for both the notified and the released variety. Opal green tag is given to this. Okay, now the next is the registered seeds. Registered seeds is derived from the foundation seeds to maintain its purity and identity. Okay, purple tag is associated with this. And now, when I say the release variety, don't get confused with the notified variety. Release variety means once the breeder identifies a superior variety, okay, in its place in his farm, then he will send that for multi-location trials and for advanced varietal trials and for release of that variety. Okay, so once the central subcommittee or the state varietal release committee, wherever you want to produce it, whether in the national level or in the state level, once the committee approves, then that becomes a release variety. Okay, and the notified variety is after the variety is released, the government of India will notify each of the zones for seed multiplication and distribution of the variety to the farmers and to the common people. Okay, this is the meaning of the notified variety. Okay, there are other three terms that are associated with this again, the extant variety. So extant variety means any variety that is notified under Section 5 of the Seed Act 1966, or it can be a farmer's variety, it can be a variety that is in public domain, or it can be a variety of public knowledge. Okay, essentially derived variety means essentially derived from an initial variety. So it is derived from an initial variety, encompassing all essential traits that is present in the initial variety, except for one or two traits that have been modified or that has been corrected. Okay, so this is what essentially derived variety means. Farmer's variety is a variety which is cultivated and involved by the farmers. It can be a wild relative, it can be land raised. Okay, that is comes under ITK of the farmer. We have something known as seed multiplication rate and seed replacement rate also. Seed multiplication rate means how much each seed sown is, each seed sown is producing. Okay, so this will show the SMR is usually calculated by the formula seed yield divided by seed rate. Okay, the next is seed replacement rate. Seed replacement rate means out of the total area of the crop that is calculated, how much of those area is governed by the quality seeds. Okay, so this again is an indicative of the quality seeds, how much farmers are using up the quality seeds, or how much of the quality seed is reaching the farmer. Okay, so this SSR will give an indication of that. Please try and remember the SMR and SSR of the important crops, okay, the major crops that I have listed here, and you may find additional SMR and SSR for other crops also in other textbooks. So please kindly refer to that because this is also important. And this is the isolation distance, okay, given for self-pollinated crop and the cross-pollinated crop. So kindly refer to this. Now, the next is heterosis. Okay, so heterosis means it is the superiority of the F1 progeny over its parents. Okay, so heterosis, when we say the term heterosis, it usually means we usually take it in a positive sense. Okay, so usually in terms of yield and in terms of productivity. Okay, so it was discovered, the concept was first came up by Shell in 1940. There is another term that is associated with this. It is called the luxuriance. Okay, luxuriance is also similar to heterosis in the sense that it will increase the vigor and size of the F1. Okay, but the main thing that differentiates between these two terms is the reproductive ability. Okay, so in heterosis, the crop will be completely fertile and it will have a reproductive ability. Okay, but in luxuriance, even though it is giving a larger seed size or larger fruit size, or the leaf size is bigger, still it will be completely sterile or it will be poorly fertile. Okay. Now, inbreeding means it is a mating between individuals related by descent or ancestry. Okay, so there are a few terms associated with this that you need to know. Inbreeding depression means over the years of inbreeding, seed mating or self mating over the years, the vigor and the fertility of the inbred will reduce. Okay, so this is known as inbreeding depression. So there are four degrees of inbreeding depression. The first is high inbreeding depression. So high inbreeding depression, what will cause this is selfing will produce the lethal types. Okay, so if a selfing will expose a lethal gene or a lethal allele, then that will completely destroy the plant or the carrier or the organism. Okay, so only very few lines will remain, will be maintained up to third or fourth generation because most of the progeny will die. Okay, and the yield will also be comparatively very less as compared to open pollinated varieties. It will be less than 25%. Okay, so some of the famous example for the high inbreeding depression is alfalfa and carrot. Okay, moderate inbreeding depression, again this will also be lethal, but it will also produce some kind of sublethal types. Okay, so some organism, more organisms will survive in moderate inbreeding depression. The yield will be 50% of the open pollinated variety. Okay, so some of the famous examples are maize, sorghum, and bajra. Low inbreeding depression. Low inbreeding depression means a small proportion of the progeny will show inbreeding depression symptoms. Okay, there will be loss in vigor and in fertility. It will also be reduced, but the plant will not. Most of the plants will survive, and it will yield as much as the open pollinated variety. Okay, and some of the examples are onions, cocoa peats, rye, and sunflower. Lack of inbreeding depression will be in the self-pollinated species because they are already self-mating or they are already seed mating. So this inbreeding depression will not be seen. Okay, in self-pollinated species. Now, again, there are three kinds of heterosis. The first is the average heterosis or your relative heterosis. This means that your F1 progeny, how far it is performing well over the average of the two parents. Okay, so this is estimated over the average of the two parents. That is your average or relative. If your F1 performance is performing better than your better parent, that is known as your heterobeltosis. Okay, better parent means you have two parents A and B. Okay, so A is giving you a yield of around 40 kgs and B is giving you a yield of around 50 kgs. So if you make crosses between the two and F1 is giving you a yield of around say 55 or 60 kgs, and that means it is better than both the parents. It is performing better even than parent B. Okay, so that means it is estimated over the better parent. So this is heterobeltosis. Centered heterosis means how much your F1 is performing better over the standard or national check or national variety. Okay, this is of importance for a breeder. So that is why it is known as economic heterosis, because when a breeder wants to release a variety or go for release of a variety, he will always check how much that variety is performing better over the standard variety or a national check or a state check. Okay, so this calculation will always be made. Okay, so that is why this standard heterosis or economic heterosis is very important for a breeder, and this is usually considered when a breeder wants to go for release of a variety. Now, there are three genetic bases of heterosis. The first is the dominance hypothesis, second is overdominance, and third is epistatic hypothesis. Okay, so the dominance hypothesis was given by Davenport, and it was later on expanded by Keeble and Filio. According to this hypothesis, the heterosis is, or the superior performance of an F1 over its parents is because of the dominant allele, okay, masking over its recessive allele. So if allele is the superior allele and the recessive allele is the harmful allele. Okay, so the dominant allele will mask over the harmful expression of the recessive allele, and this will cause heterosis. Okay, this is one of the most widely accepted theory among the three. Okay, and the overdominance hypothesis is given by Fisher and again expanded by East and Shull. Okay, so according to this theory, it is not because of the dominance of it is not because of the masking effect of a dominant allele over a recessive allele, but it is because of the state of heterozygosity itself. That means It is because, not because A is dominant over small a, but it is because capital A and small a, they are both present in a heterozygous state. Okay, so according to this hypothesis, heterosis is because of that. Okay, now if you see the picture here, I mean the picture that I have, the figure that I have given here, impred 1 and impred 2, okay, in cross 1. So if you see the impred 1 and impred 2 in cross 1, it means you see the original genetic constitution of both the imbreds. Okay, so in this, their F1 is completely heterozygous, plus the dominant allele is masking over the recessive allele. Okay, so that means in this case, both the hypotheses are working, both the hypotheses are acceptable. Okay, in cross 2, the original composition or the genetic constitution of each impred, parental impreds are given, and the F1 have 2 heterozygotes, but C and D, they are both in recessive form. Okay, recessive homozygote form. So there, according to the dominance hypothesis, where there is a masking effect, there will be no heterosis. Okay, according to dominance hypothesis. But according to the over-dominance hypothesis, because F1 is in a heterotic condition, that is why it will have heterosis. Okay. Now in cross third, in the third cross, your F1 will have 2 completely dominant homozygotes and 2 completely recessive homozygotes. Okay, so in this case, both the heterosis will not be present. Third hypothesis is epistasis hypothesis given by Gowen. In this, it means that influence of one locus on the expression of the another locus, this will cause heterosis. Okay, but again, all the heterotic crosses do not show epistasis, and all the crosses that are epistatic are not heterotic. Okay, so again, this epistasis heterosis is acceptable only to a certain extent. So the most widely accepted hypothesis is dominant hypothesis, but again, overdominance and epistasis hypothesis are also accepted to some extent. Okay, so I have just attached this page just to show, because they keep asking these questions like what is the genetic composition or genetic constitution of a hybrid or a composite or synthetics or a pure line. Okay, so you please try and remember the genetic composition of each of these. Okay, first of all, try to understand the terms. What does hybrid means? What does composite or synthetic means? What does pure line means? Inbred means? Okay, so try and understand this. Then you will understand why their genetic composition is, for example, for hybrid, why the genetic composition is heterozygous and homogeneous. Okay, so for hybrid, it is heterozygous and homogeneous because hybrid, as you know, they derive the alleles from 2 different parents. So they will be in a heterozygous form. Okay, different alleles from different parents, so they will be in a heterozygous form. But they will be heterozygous only for that particular gene or for that particular trait, because hybrid is usually a cross that is made between 2 impreds. Okay, so in other locus, they will be similar. Okay, they will have similar maturity dates, or they'll have similar seed shape or flower color or plant height. Okay, all of this will be similar except for the trait that you are improving. Okay, so that is why this is heterozygous within its gender constitution, but the population is homogeneous. Okay, so again, you need to remember all of this. Why it is heterozygous, why it is homogeneous, why it is heterogeneous. Okay, you will only understand this after you understand each specific term. Okay, and then I have also attached a picture here where the source of cytoplasmic male sterility is given for each of the crops. So for rice, it is wild abortive. Okay, for cotton, it is Gossypium harknessi. For maize, it is CMS CTS, and for sorghum, it is Kaffir 60. So all of this is very, very important. Please try to remember all this. Okay. Some of the books I would like you to refer are the For plant breeding, the ones that are marked in red, they are very, very important books. So please go through these books. Okay, so the ones that are marked in black color, you need not, if you have time, then you can go through, but you need not essentially incorporate. Okay, so for your level and for your exam, I think the one that are marked in red should be sufficient. So fundamentals of genetics or genetics, BD Singh, plant breeding by BD Singh, cytogenetics by PK Gupta, okay, and all these books. So for those students who have the habit of solving objective first, I would, and for every one of you, I would like to suggest you all not to solve objective first. Please read the subject material first. Please go through each of the concept and theories and the lessons first, then solve the objective after you're done with all of this, okay? Because then it'll be easier for you also to understand, because if you just solve the objective, then you're just memorizing the term or the concept, okay? So if you, instead of memorizing, if you understand the concept, it'll be easier for you to solve the objective also. So please go through the subject first, read the subject first, and then you can solve the objective, okay? There are books that I have given for plant breeding, for microbiology, science and technology, plant pathology, and plant genetic resources and general agriculture. Okay, so for plant genetic resources, if you have time, you may study. Otherwise, I will also send you some of the soft copies that I have, some materials I have in soft copy for plant genetic resources. So I will share that with you as well. Yeah. And I wanted to show. Yeah, I have also included some of the frequently asked questions over the last 10 years. Okay, so please go through this. Quality seeds, I have already discussed with you. The scientific names and the genome and the families of each of the major crops. Okay, and then the varieties that are derived from somoclonal variation. Okay, then from protoplast fusion. So all of the varieties that have developed, and also study the hybridization of the sugarcane or hybrid millets, then hybrid cotton, okay, then the green revolution. All of this, please do not miss out, please study. And then the GM crops, different GM crops, BT cotton, BT brinjal, GM mustard, golden rice, okay. And then, yes, the genetic composition, I've already discussed in the last slide. Genetic composition of hybrids, composites, synthetics, lines. Okay, all of this you can study. And then the latest development in plant breeding and genetics, like CRISPR-Cas, speed breeding. Okay, again, you need not see the entirety. Just study the concept of what it is and who came up or who developed the method or the program. Okay, so you need to study that. And yeah, so most commonly asked questions I've included from all the sections, from plant breeding, from plant pathology, from other sections, from crop physiology also. Okay. Okay, so some of the tips, again, everybody has a different style of studying. So stay focused, stay consistent, and you might study for a few hours, but you might have a quality study. You might study and sit in front of the computer your whole day, but you might not get anything inside your, inside your head. So try and do a qualitative study instead of a quantitative one, and make your own notes. And again, revise again and again and again, because there are terminologies, there are concepts, there are theories, there are years, scientific names that you need to remember. So you need to revise it again and again and again. Okay. Again, stick to the syllabus. If you have time, you can venture more, but stick to the syllabus. Yeah. And for your examination, approximately spend 1.5 minutes for each question. This is just tentative time. And again, during examination, I understand because of the pressure, it's not possible to strictly, calculate how much time you have to spend in each question. But if as much as possible, try and spend 1.5 minute in each question and attempt the question that you know first. So you can finish off all the questions that you are sure of, attempt that first, and you can always come back to the other questions that you're not sure of. Okay, and if you're not sure about some questions, if you're completely not sure, then please don't attempt it, because then you will invite unnecessary negative marking. For some questions you might get confused, so for those questions you can take few chances, okay, but try and avoid questions if you do not know at all. Okay, please don't make like futile attempts. Okay, so yeah, yeah, it is just a phase. You will all come out victorious. Just stay consistent, just prepare well, hydrate yourself, and take proper care of your health also, because that is of utmost importance right now. Maintain your immunity, and yes, focus on your preparation and stay consistent. Thank you.
Yeah, thank you so much, Dr. Philanim, and such a wonderfully you have covered the syllabus, starting with the syllabus, and then you have nicely covered all these what is heteroploidy, heterosis, and all these with a very good examples, and the students could observe. And some of them, and lastly you could give some of important questions, and you have stressed on it, and that also given a lot of hopes for the students to which line they can prepare it to crack this JRF examination. The last one is not the least, but the tips you have given them, nicely, like how to focus and study in this pandemic time, quality time to spend for the studies, nicely organized. Thank you so much. We would like to thank from our college on behalf of our dean, Dr. B.N. Hazarika, and he's a nodal officer for this, as a associate professor in plant pathology, and then as associate nodal officer in IDP Nahe and IDC cell, Dr. Nansal Bhutia, and then Dr. Gita Rani. They are also the coordinator for this JRF exam. And really, we thank you for your valuable preparation to arrange it nicely and present to the students. And due to some fall short of network and connectivity problem, and many of them watching online, I could see that. I will send the link to you also, and you can also watch this. And it is recorded, and they can access to your information anytime. So finally, thank you so much. And so we will come back to you. So for the further one or two classes. Okay. So thank you so much. Thank you, dear students, for your consistent, consistently you are attending these classes. And tomorrow also we have a class for the soil science. So I would like to request students to join the tomorrow, same time, 11 a.m. Thank you so much. Till then, thank you. Bye-bye.